كوبي عندك
فين هي فيها؟ كاين هي الكلي
شي خاصكم تكونوا كتصوروا ماشي مزيان
تعطينا غير الباوربوينت
الباوربوينت ديالها بريزونطاسيون ديالها باردون خليها غادي ستريمين خصنا نديرو ستريمين دابا ستريمين خاص يبدا
بريزونطاسيون
laquelle
July presentation July 20.
هاد لاست وان هي اللي كاينه بوجوردي
هي
عندك كوبي عندك ديروا 16 نوفمبر حل باوربوينت حل كلشي vous avez un micro ici vous voulez vous asseoir
Non, je vais te dire.
ok vous avez un micro main et après il y a le cliqueur ça c'est le pointeur laser il y a le cliqueur
Un micro خصني
oui bien sûr je t'ai dit on a fait streaming là parce qu'il y a beaucoup de monde qui il faut vraiment rapprocher le micro
c'est la présentation
1 2 test
1 2 تيست 1 2 تيست رشيد كتسمعني في الميكرو كتسمعني في يوتيوب رشيد 1 2 تيست 1 2 تيست رشيد كتسمعني تماك ياك صافي مزيان
Un de test. 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 Un de test.
هدر قريب لي جون في يوتيوب باش يسمعوك علاش درت في يوتيوب انا ما قريتش YouTube, j'ai pas YouTube. Ils ont dit, je prends des photos YouTube, je n'ai pas de YouTube. Pas de YouTube.
دونك شنو بلا ما نديرو ستريمينغ
Bien, merci. En votre nom à tous et au nom de l'université, je tiens à remercier vivement Dr Sloa Khargoun, donc de l'université de McGill, qui est professeur en biotechnologie alimentaire. Et donc, vous avez eu son CV résumé. Et qui est en même temps vice-doyenne de la faculté d'agriculture. Donc on a le plaisir de l'accueillir pour nous parler un peu de ce qu'elle fait en particulier. On l'a invité pour deux choses. D'abord pour nous parler de l'université de McGill et pour essayer de voir qu'est-ce qu'on peut faire ensemble avec McGill University. Voilà. Merci, docteur.
Merci beaucoup. Merci pour cette introduction. Et merci de m'avoir accueilli. Vraiment, j'étais chaleureusement accueillie. Merci Obeid pour faciliter ça. Je suis impressionnée par le site. C'est vraiment un très bon site. Et puis, Inch'Allah, il se dahar plus qu'il le faut, Inch'Allah. Je vais donner ma présentation en anglais, juste pour trouver mes mots. Donc, ça va être plus facile pour moi. Donc, je viens de l'université de McGill. So we are located in Montreal Island. It's a one university actually, with two campuses. We have one at the center of of Montreal, and the other one is at the West Island. So my faculty actually is at the West Island of Montreal here, the green. And really, we all the time try as much as possible to keep our green color because it's agriculture based. McDonald campus, which hosts my faculty, is extended around 650 hectares, and all the sites for research is around this is at the faculty. And I would say this is one of the advantages of my faculty as compared to the other ones in Canada, is that all the experimental sites are located in the same campus. So we have a farm, and it's the last farm that is available in Montreal Island. So we have cow, we produce milk, we produce many other produces, and we make use of it as well. We just here to highlight some of the sites. So we have a dairy farm, as I mentioned, horticulture center, and few other ones, seed farm, arboretum. This is a very important, I mean, it's not important, it's a nice place that I like to walk into it. It's a forest, it's a small forest that has many species of the trees. Just showing here a landscape of my faculty. Each faculty has a mission or has a mission. So our mission, I can say that in this sentence. So the Faculty of Agriculture and Environmental Sciences is committed to excellence in teaching, research, and service to ensure that humanity's present and future food and health and natural resource needs are met while protecting the environments. So if you see from here, we have some keywords. So it's about food. Food is important, health. We know that there is a relation about food and health, and I'm actually working at this interface. I will talk about it more in details. And natural resource as well. We have a very important department there where we deal with many natural resources from different perspectives. And of course, the environment. So those are all the ecosystem that we need. We cannot more talk about each one of these components separately. We have to look at them on this globality because it's important. So we have 6 academic units, including the Institute of Parasitology. And what is interesting is that we cover the chain lengths going from plant science, animal science, to more natural resources, sciences, and agriculture economics, bioresource engineering, School of Human Nutrition. So we have a school where we train nutritionists and dietitians as well. And we have the food science agriculture. So my host department is from food science. So I'm a food biotechnologist and I'm working at the food science. Now, what we do as well is a centers and network. And as I mentioned to a few of you, so these centers are virtual, but they're important because they kind of host many expertise. And they have been built up depending on the need of the governments. So I want to highlight a few of them. So McGill Institute for Global Food Security, which is one of them that was maybe put in place 10 years ago, and that's because of need of food security. Now we talk about nutrition security. We don't want only to have food available for people, but we want as well nutrition security, to have food that is healthier for the consumers. We have as well the Biofuel Net. I'm going to talk about that. It's a center that has been funded by the government for biofuel. We have the Brace Center for Water Resource Management. I know that few of you, they are interested in water management. And it does gather many expertise related to the water management. And other ones, which is, this one is a very interesting one. I'm going to mention a few things about it. It's McGill Center for Convergence of Health and Economics. And we are doing that in collaboration of the Faculty of Management at McGill. So the Faculty of Management will look at how the economic aspects, but we want to include the health in the economic aspects because we don't want more to deal with economic without considering the health from its globality. So my faculty has 90 tenure track faculty members, like 90 profs. Here just some of the chairs and scholars and endowments. What is particular is that since 2005, more than 44 tenure track professors were hired. So we're quite young faculty and we like that and it's good for us. So it's like most of the staff were renewed like within the last 12 years. So what your president did ask me what's particular in your faculty. I mean, MAC, I consider it as a hub for research and innovation of sustainable systems. So it's not only agriculture, but it's also ecosystem, looking at the globality of the ecosystem, the landscaping, the urban, water management, plant, food, and nutrition. This is really our hub. This is our key areas where we work on it. But if we look at it from a research perspective as an associate dean, so we have 3 main focuses that we can put them in under these 3 umbrellas here showing. So we have food, nutrition, and health. We look at that from different angles, from production to the end use of the products. And I'm going to give a concrete example because Now, we do actually, we do grow and we do produce many goods, but we do not consider nutrition from the beginning sometimes, and most of the time I would say. But if we do consider nutrition from the beginning and looking at the nutritional value of the food products, we make better use of the food products. And I will give an example of quinoa. It's a collaboration with ICBA and, in Dubai, in which we have looked at the quinoa production not only from yield production, from yield perspective, but also from nutritional perspective. We have the bioscience, biofuel, bioproducts, and we have the environment of ecosystems. This is our main focuses. If I want to put them under some specific areas, few of them are mentioned here. So either nutraceutical and functional food, detection of contaminants and toxicants, dairy herd genetic health management. Biofuel is going from plant genomic breeding solution to antibiotic resistance in livestock. That has been a main issue. We are looking for other alternatives. Biofuel, alternative energy resources, sustainable agriculture, soil fertility, water quality, and the natural resources and wildlife as well. So, sustainable agriculture systems, I selected this one. I assumed that it's something that may be an interest because both between your institution and mine. And if we think about sustainable agriculture system, we have to look at it that something that's really merged, and we need many expertise. And it's not more just the traditional way how you're looking to that. We're looking to the natural resource management. It go beyond that. Now we're talking about precision agriculture, which is something very I found myself very interesting. It's to be more like trying to model it or trying to more target the need of the soil and doing that on small scale. It's going to more electromechanical control system, irrigation and drainage, to more sophisticated science where we are looking to the epigenetics, abiotic stress, plant gene regulation, bioinformatics. We talk about the endophytes, and we talk about the scanning, trying to get as much information from the CT scanning or other type of scanning without having, or keeping the plant in life. Then we can get more information from the plants. So the other initiative that I wanted to mention is the CRIBS, which is a network for Canadian research, integration, and innovation in biofuel sustainability. It's a center, it's a network that was created and led by our faculty, and it focuses on the development of sustainable and renewable energy, and that will not compete with the food production and will not harm the environment. And it has been focuses on 4 main areas: feedstocks, conversion, use, and socioeconomic. And the way how we did implement that, it's having these 3 platforms that they are more in the vertical way, and we have done it with region. That's something very important to respect. So here is the West and the Prairie, Central. This is the region from Canada. So because we can't implement the same structure in all regions. We have to look at what exists in the region and what are their needs. So the way how we have implemented that is to look into each region, but the main areas are the same, but they may have to be tailored depending on the regions. And of course, the social, economic, and environmental sustainability is more in the horizontal way than in the vertical way. Ecological engineering and ecosystem management. So during the last 10 years, we hired many new profs that work on these areas because we know the importance, climate change. What is particular and what my colleagues are trying to do is to link the human and the ecosystem together. Because sometimes we look at the practices, but we don't look at the human in itself, which is important, or we don't look at the ecosystem. So it's trying to really consider the human as a particular component when we look at the ecosystem, because at the end it's the human practices that change all this management. We have created McGill Institute for Global Food Security, and it addresses agriculture, nutrition, and other pressing human need, environmental in many in India, Sub-Saharan Africa, Colombia, Kenya, and Caribbean, and we were able to secure many funding. to reduce the food insecurity, the malnutrition. This is some example of some projects that they have been funded. The Global Affairs Canada is the one that I had mentioned to you that we are now targeting Senegal, but recently we obtained 46 million in partnership with Ethiopia, Mozambique, Malawi, Zambia. So there is a lot to be done in Africa, and I think your university is really located, or it's positioned in a good position to be able to contribute to food security in Africa, in generally speaking, if that's something very interesting. Look into different types depending on the area. So the Institute of McGill Global Food Security, I would highly recommend you please go and visit the website. The director, Hugo, is a very, very nice researcher working on that. And again, it's a very social institution that you can be part of it. They can share information with you. And we organize a conference each year that helps to do more networking. We also, so this is coming from an area where we cultivate, we process, and we look at the quality across the chain links. So we have expertise at different levels, going from animal, plant, agriculture to developing innovative products, to processing them, and to looking at the quality and safety of the food products. To mention another opportunity that, or I would say it's another initiative that we have, it's the omic approach. So we talk about data, so many data that can be generated, but what is important as well is how to generate this data. And omic approach, genomics, we started with gene. So genomics, we are doing gene analysis of many strain, of many other things. We can get plenty information from that. But the omics approach is not only now looking at it from a gene perspective, we can also talk about foodomics. And I'm sure there will be agronomics. So its omics approach is trying to get many information and even soilomics, why not? So it's trying to get all this data and then trying to analyze it in a way that we can get so many information. So we have this project that was funded recently by Genome Canada and all these partners, in which we are looking to the omics approach to ensure safety and reducing the economic burden of salmonellas, where from farm to the table, we try to get different types of salmonella and try to analyze them from an omics perspective. Management of environmental chemicals as well. So this is just showing an example of another initiative, which is an important one, that was funded by Genome Canada and some industrial partners, in which we try to identify some ecotox chips that are abundant and that help to assess the toxicity in the environment. It's really the environment that we are targeting here, and that help us to first to manage that, because at the end we want to be able to manage it. But to be able to manage it, sometimes we need some tests that are rapid and reliable as well. So toxicity, we are looking at it from a microbial perspective, but we're looking at it from a chemical perspective as well. Oh. So that's my last, that's another one that I'm personally leading. So here we have our Dean, Anya Gutman. We have the Ministry of Quebec, Monsieur Lessard, and we have our VP office. So recently we got funding from the government that help us to put a consortium de recherche pré-compétitive sur la transformation alimentaire. So this is the last one, the very recent one. So the idea is to create a platform where we'll have so many partners around its table. Is one of my goals in my mandate as an associate dean is how to bring industry into my faculty and to get more projects with the industry. And we have done so many workshops, and that was the result of creation of this workshop, is to have this consortium. And also to talk, I don't know how but we talk a lot about open innovation, and it's something that we're trying to move into it. It's instead of having each industry doing their own research, or trying to be more open innovation, because there is some ground of interest between all these big industries that they should share resources, because we want really to share more resources, and more importantly that these resources can be also shared by small companies, which we do have here in Morocco as well. We value entrepreneurship spirit. this is one thing that I wanted to mention. So just simply by creating a kind of competition for students to come up with the new ideas, with the new tool, this is really, it does stimulate the students, but also the prof as well. So the entrepreneurial spirit, it doesn't cost much to the faculty, but really help to build a very nice environment for the students. This is through partnership, so something that I mentioned. So we try to create a pilot plant for renting. This is one of the food lab that we rent. So that's create, sometimes companies will come to rent facilities, and then the first time they just rent it, the second time they create more partnership. And one thing is that we are targeting, this is our goal, is to create a residential college spirit. And I think the environment that you have here is the best to create that, is to be able to create a unique experience for the students, having teaching, research, and living in one place, providing integrated student service. So we put a lot of effort trying to improve and foster the residential college spirit, because we think it's important for our faculty and for the environment. We try as well to reach the community. So we talk about how can we reach the community, and one way we do that is trying to create some engagement center. This is one of the last projects that we have as an engagement center, is to have here a farm opportunity where people from community can come visit and see what even some of the prof, they can expose the research that they are doing, and it's really basic agriculture and create community, which is important, even more than what we think. Okay, so now I'm going to move to the right topic, which is research, which is Agri-Food Innovation Platform, Challenges and Potential. So I will be focusing mainly on my program, research program. I like this saying, this is the one of Charles Darwin. So it's not the strongest of the species that survives, nor the most intelligent, but the one most responsive to the changes. And I believe that's really the real. So it's not to be able, it's really to be able to respond to the change. And here we are talking about the change of the people, change of population, change of the society, and so on. So if we look at the agri-food trends, and we look at the change that we are looking at, now, so many years ago, we were having a product-driven platform. So the product that was driven, the platform, is a type of the product. Now it's not more the case. It's more function-driven platform. So it's really what are the function, what this food is going to bring me as a health benefits. And the way how I see that, I see it as a way that it does offer us the agri-food sector. When I talk about the agri-food sector, it's not only the food transformation industry, but also the agriculture. It does give us an opportunity to become the leader for wellness management and disease prevention, like such the pharmaceutical company that they are more for disease management, this one with wellness management. So if we look at this platform, it's a platform that did evolve through many generations. It's not new. First, we try to fortify the foods. Then after that, we're moving to the second generation, where we're trying to make food with a single health benefit. I'm going to give some example. Now we're moving to the third generation, where we're talking about multiple health benefits. And I discussed with your colleague that what is important is the synergistic interaction. So more we understand how these bioactive molecules are metabolized, there is a synergistic there that we need to take advantage of that. And of course, in the future, we're talking about nutrigenomics, and I have one colleague working on that, where they're trying to link the gene, the human gene, based on the diet. So depending on what is your gene, then he can tell you what you are susceptible to have as a disease, and then you can have the right diets that prevent you from having this disease, actually. Why we're having this move? Because people are beginning to associate healthy diets as cheaper than going to the doctor. And this is the reality. So, and we feel like as an agri-food sector, we need to have this impact, and it's important impact. So one initiative that I'm working on, and this is, I would have to say, this initiative is done at the national level. There are many, many partners at the national level, and it's my faculty and the faculty of management that are involved, is to try to create a platform for innovation, where we're not only looking to which product. No, we have to look at the health of the economy, the health of the environment, and the health of the people, and to come up with to come up with the platform that can help to produce, promote, and consume food that sustain the health of the people, health of the environment, and health of the economy. It's all of them to look at them at the same time. And of course, this is driven by the citizen, consumer, and passion. So food people that they want, food the farmer, and the value chain. The value chain is very important. And we looked at that, it's very important. It's not only to grow producers, but you have also to ensure the value chain, that the value chain is there. So this is the strategic role of each one of those. So the current issues here is that the chronic diseases, healthcare cost is so high. We have climate change, we have soil organic matter, we have water quality, we have carbon footprints, economic uncertainty as well. So this is what we have as a challenge. As a person, there is other challenges here or need as a society as well. So what can we do? How can we create our food that we need in the future, and how can we do that? So we created this platform. It's really a holistic view in which we looked at all the different whole ecosystem and many players. So we can see here that we need players at different levels. And we try to create a relationship between all these players, how to connect them. So to have food is medicine, food is life, and food is sustainability. And at the end, research and innovation should be driven by all this holistic relationship instead of having like a silo, each one of these components from this whole ecosystem working on their own. But there is difficulties that we did identify here. So it's fragmented group, the efforts that become more powerful and sustainable when they are integrated. So we're working on that. We need to speed, we need to duplicate the effort. So there is a shift in requirements in the innovation. And there is also a policy that should follow, because we understood that even though we want to incorporate any platform, we need the support of the governments and policy. Because if the government provides support for the agri-food sector that produce healthy food, all those industry will go and will produce them. If there is no benefit of doing it, they won't do it. And the government has to see the benefit as at the end. It's like they will prevent many diseases. Okay, so myself, what I do as research. I develop an innovative strategy for synthesizing functional ingredient and nutraceuticals. We have been implementing many ways of doing that, creating values, designing new molecules, maximizing synthesis, improving functionality and bioavailability. I'm going to try to give example of many projects in which we have used each of this approach. At the end, I'm working at the interface between biocatalysis, enzyme technology, and food chemistry. So I consider myself as a chemist, and I think chemistry is important because it's the base, because it's the chemistry of the food that will determine the properties of the food, the health properties, and even the techno-functional properties of the food. And also, I'm using some nanotechnology to improve the biocatalytic systems and also as a delivering system as well. So why biocatalysis is a subfield of biotechnology? Simply because it does help to respond to many critical aspects in preparing ingredients. When we prepare an ingredient using biocatalysis, we can have a natural label. And in Canada, it's so important, the natural label. So natural label, clean label, that's what the consumer are looking for. And also the quality, and it's environmentally friendly. Okay, so the first project that I would like to mention is about potato. We eat a lot of potatoes here, right? We never think that potato has protein, but potato has protein and has a very nice protein. Potato has polysaccharide and has a very unique polysaccharide. And that's what I try to do through this project. It's a project that has been funded by MAPAC. It's more like a provincial funding. We looked at the potato protein, potato polysaccharide, and we tried to combine them through glycation, Maillard reaction, or through glycosylation. Let's start with the pectic polysaccharide. So pectic polysaccharide is a pectin. I won't go in detail, technical detail. I will just go very fast. So it's a pectin. It's very complex polysaccharide. It has very different groups. And if we wanted to to compare the pectin from orange, from apple, from sugar beet, the one from potato, it has a very high neutral sugar, and it has a low degree of methylation and high degree of acetylation. So it cannot form a gel. I mean, the pectin from apple and orange is what we use to make jam, because it's from a gel. It provides very nice gelling ability. But the one from the potato does not form a gel, so it didn't have any really real use. But we have, actually, it has some very interesting use. We can use it as a source of dietary fiber. We can use it to prevent tumor growth. And there is, and that's something we have done it in collaboration with the lab in Cornell University, and we found that the galactose-rich ranogalactonan from potato can prevent the free cancer cell from binding together, and also it can have a prebiotic activity and anti-adhesive activity as well. So the first thing that we had looked at it is how to isolate this pectin from potato. So we looked at it in a different way, and as being an enzymologist, so I wanted to use enzyme, and I have proven that by using an enzyme, I can get my polysaccharide without having any debranching, because the challenge is how to get it intact. I don't want to cut it in small pieces. I wanted to get it intact as much as possible. So if I compare other type of extraction, like alkaline extraction or others, with enzymatic extraction, I was able to get my polysaccharide in good shape. We optimized the process. We did a modeling to do that. So this is help us to determine, not only to determine the optimal conditions, but also to try to target the structure of the final product. So depending what application are you looking for, so you will need a polysaccharide with some specific structure, and the modeling helps to do that. So it helps really to determine the condition to synthesize a targeted polysaccharide. We were interested in generating prebiotics from this rhamnogalacturonan, and we identified the enzymes that helps to do that. And the prebiotics that we were targeting is the galacto-oligosaccharide and the arabino-oligosaccharide. And of course, when we talk about prebiotics, what they do, prebiotics, they stimulate the good bacteria that we have already in our colon. And to be able to do that, so we used many enzymes. We identified the good one. We looked at their efficiency in terms of yield. We looked at their efficiency in terms of number of sugar that we have, that our polysaccharide has, and also which type of one. So our target was to get the galacto-oligosaccharide. So anything that was here scratched in red, it's because it doesn't have high galactose that we had targeted. And then we looked at their fermentability. So we tried to mimic like our colon. So we got the host bacteria from a human, and we had immobilized it and feed it with our polysaccharide, and looked at how the microbial population, the microbiome, it does vary, and to calculate the prebiotic effect. We did also the digestibility using a TIM system, because the idea is that we don't want our polysaccharide to be digested. We want it to reach the colon and then to be fermented by our beneficial bacteria. So we wanted to make sure that it won't be digested. So it's just to show you some of the results. So here does show that our oligomer and polysaccharide are not digested, so it's a good news, so we can go further. And then we looked at, because in our colon, we have more than 3,000 bacteria that they are already hosting here. But we wanted to have them to be fermented, the bacteria will be using them as a carbon source, but we want the beneficial bacteria, the lactic acid bacteria, not the bad one. So here, as you can see, we have a decrease in the growth of the Clostridium group, which is a bad bacteria. Bifidobacterium, we have an increase, lactobacillus, and we compared that with the standard. So we were very happy with the results. And when they are fermented, they produce fatty acid. So we looked at that, and we can look at the total fatty acid here. The potato protein. So potato protein, how many of you think that potato has protein? Not many, yeah. So if we compare how much protein can we get by hectare, you can see here it's quite competitive than rice. It's 0.3 ton by hectare. If we compare it, it's even higher than corn. So in terms of content, it's not so high, but when you look at the efficiency, it's really high. And there is only one company in Belgium. No, it's not in Belgium. Anyway, I forgot where, that they're producing potato protein. But there is a big potential on this protein here. And why there is a big potential? Because looking here at the essential amino acid, it has high lysine, high methionine, which we are looking for. Remember, when we talk about protein, we have to talk about the quality of protein as well. So we are looking for having high essential amino acid. And what we have done, we have developed an enzymatic approach that helps us to isolate this potato protein. And the enzymatic approach is based on using diverse enzymes that they're going to degrade all the polymers surrounding the protein. And then after that, the protein, they are going to be released. And we get our protein. So when we did that, so this is different multi-enzymatic product, we were able to recover a high yield, but also we were able to fractionate, because when we think about potato protein, there are two types, patatin and protease inhibitors. Protease inhibitors, we're using them for satiety effect. So when people, they want to use diets, they can take protease inhibitors and then really give them a satiety effect. And patatin has other important beneficial properties. Now, as many of you know, that when we deal with protein, they are thermolabeled protein because they can be very sensitive to any way how we isolate them. But as was our target, to isolate them in a native form and to preserve them as much as possible, and to be able to determine if we have isolate them in native form. So we look at some of the techno-functional properties and compare it with other methods like foaming and emulsifying. We looked at as well some protease inhibitor activity, lipid acylate hydrolase activity. I won't go in details. We do also a modification of protein, and we can do that by enzymatic glycosylation or non-enzymatic glycation. Glycation, which is a natural reaction, and we have tried different glycosylation, different glycation, using different inhibitors. So why would we modify proteins? We did modify the proteins for many reasons. One of them, to make them more resistant, to decrease the allergenicity, to improve some techno-functional properties. And in my lab, we developed this expertise of modifying the proteins, characterizing them, and looking some of the techno-functional properties. Like here, an example, looking to the allergenicity. So currently, we are working on gluten. So we know gluten can lead to celiac, and we are trying to modify the gluten in a way to decrease the allergenicity, and we are doing it also with peanut butter. So the idea is to modify the protein without having to remove it, just to modify it, and the allergenicity will decrease of the proteins. Okay, the other project that I want to talk about is designing new molecule. So now, as we understand, sorry, I'm going fast. Okay, as we understand how the molecules are metabolized, we have better understanding what we need. And here our target is prebiotics. Prebiotics are selectively fermented ingredients that help to change the composition, the activity of the gastrointestinal microbiota, and they provide many health benefits. And even they will say that our colon is our brain is the brain of our health. And I do believe that, because many studies, they have linked the host to the diabetes, to the many diseases. So if we are able to keep a good GOST, a good microbiota, we are able to prevent many diseases. So the idea here was to have a good understanding of how they are metabolized. We have pathogenic bacteria, we have good bacteria. It's trying to decrease this as much as possible and to promote the good bacteria here. So if we look at the prebiotic, the challenges that we have is that they're not selective. So what I mean by that, I want them to be used only by the good bacteria. I don't want them to be even used by the bad bacteria. That's the challenge. We want them as well to increase the colonic persistence. We don't want them to be metabolized at the proximal region of the colon here, but we want them to be metabolized at the distal region of the colon where most of the diseases are originated. It's how to carry them to the end. And also, we want them to prevent their adhesion. So by changing the structure. So here are our targeted molecules. And what we have done substrate engineering, combine different enzymes to try to synthesize them. We are working with 11 sucrase enzyme. It's a very interesting enzyme. You just put this enzyme with sucrose, and then you get all these molecules are produced. This is wonderful. So I will just avoid some technical details. So our goal was to understand how these enzymes are working, and also what is interesting with this enzyme is that when you put sucrose and other acceptors, then you generate a panoply of products showing here. And if I take an example here of L-rose, this component here, we can find it in honey in very small amount, and it has an anti-inflammatory and antibacterial properties. But with this biocatalysis, we can produce enough to make a good use of it. So just to show you some example of the yield, so we are able to, of productivity, we're able to have a good yield of these components when you use maltose and when we use lactose. So it's here each time we're using sucrose and this one as an acceptors. We're also working on the combining different enzymes to be able to improve the yield and the panoply of the product. We do modeling. We like to do that because it's very interesting, and something easy can be done for any process that help us to understand better our processes and improve them. One thing that we are working on is how to, so I have my enzyme, but I want to immobilize the enzyme on the support and I be able to reuse it. So this is a collaboration with the lab in Spain, in which we are producing mini beads, and we try to functionalize them with different groups and immobilize enzymes on it and try to see how the efficiency of the enzyme. So just to show you an example here, when we use free enzyme, we have a yield of 61, but we are producing just 2.3 grams per liter of leaven. But just immobilizing the enzyme, we can produce we can produce 15.2 grams per liter. So how many of you have tried maple syrup? I won't ask Obaid. So maple syrup is our honey. So it's really, we produce it in Canada, in United States as well. And one of the challenges of this industry is product diversification. Because you are looking on way to increase the value of it. It's already expensive, but they're really looking for different way. How can we diversify it? There was a kind of bad publicity about high sugar products, and maple did suffer for that. So they did link sugar to many diseases, and so they wanted to find other ways how to make it, to revive this product. So I had this project with them. I had my enzyme 11 sucrase. Remember, I take my enzyme, I put it with sucrose, and then it can generate many prebiotics and many good products. So when we started that, so Quebec, we know it's produced a lot. We contain 90 to 98% of sucrose, which is the ideal enzyme for my, ideal substrate for my enzyme. So we did a trial, and we were very happy and that it did work. So when we take maple syrup with different bricks, maple has sucrose, then when we mix it, we are able to enrich maple syrup with this oligosaccharide without having really to go to the different processes. So this is one of the applications. It's stuck. I cannot move it more, no? I may need help. So here, if you can see, so we use maple with different bricks, and the idea was to look at, in the process, to look at, oh, sorry, yeah. It's just to look at which level should I incorporate my enzyme. And here you can see that we can go up to 60 bricks and it can still work. But the product, I mean, the difference is just what I'm producing oligomers, polysaccharide, or disaccharide. And we did some modeling just to see how the different parameters, enzymatic unit and reaction time, does affect the yield of the product that we are targeting. Okay, so another project that I want to mention is how to create value and increase bioavailability. So many natural components that exist, but we cannot incorporate them in many food systems because of the compatibility as well. So what we have been doing, one example here, is taking prebiotics and taking phenolic compound and trying to link them through an ester linkage. So to synthesize what we call a ferulated oligosaccharides. These ferulated oligosaccharides do exist naturally, but the amount is very, very small. So we inspire a lot from what is naturally exists, and we try to develop enzymatic processes that help us to synthesize them in good yield. So, and the challenge was to find the best biocatalysis. The media was a challenge because if you notice it, we're dealing with two substrates that they have different physicochemical properties. One is really hydrophobic and the other one is hydrophilic. And we were able to produce some of these components showing here. So I have to tell you something here. So when we look at the yield, it's quite low yield. So it can be disappointing to have this yield, right? But actually, when we have done the MS analysis, we realized that because when we did the yield, we did look at how many of the ferulated oligosaccharides are formed. But actually, we noticed that we were expecting to have ferulic acid incorporated into each monosaccharide or each dye and so on. But actually, as you can see here, we have so many ferulated group in one molecule. So the yield, it depends how we look at the yield, but the fact that we have more than one molecule of ferulic acid incorporated in the polysaccharide or disaccharide, that shows us that the yield is not too bad. We looked at the antioxidant activity, just to make sure that the antioxidant activity will not be affected by the ferulation. And we can see here that there was no effect, so we can still use this component because of the antioxidant activity. And the antioxidant activity of ferulic acid is all the time a big, it's not a big concern, but there is a big question there about how they are metabolized, do they reach the right place? So we are hoping by having a non-digestible component that they are going to carry them up to the distal region. I'm going to skip this one because maybe it's too much. How I'm doing with the timing? Okay, we try to do that. Okay. So opportunities, functional food, and I wanted to put that because I think there is a big potential here in Morocco for that. So as I mentioned, the trend is to make functional food, but this should not be at the expenses of taste. No consumer will buy a product if it's not tasty. So we still say taste is important. So we want to make product tastier but more satisfying. And when we talk about functional ingredients, there are some conventional one, probiotic, marine algae. We talk about algae today. There is a lot of potential there. What are the bioactive molecules that we can get from algae? Anthocyanin, and we have spices and herbs. Just to give you some of the traditional and non-traditional ingredients that during the last years, we have seen many products that do contain those. So we have curcumin, amla, hibiscus. We discussed about the hibiscus, lycopene, pepper, and salacia, and there are many. So just to give you some example, curcumin, turmeric is, how we call it? Kharko, that's it. So curcumin, there is a lot of products into it. So it has been proven that it can reduce cholesterol oxidation, it can really build up bad cholesterol, pro-inflammatory. And you can see here that we have a lot of products, and many of the tea bags now contain curcumin as well. We have the beta-glucan from oats as well, so we have seen during the last year many products that contain this. We have the resveratrol from crabs, so from chocolate. A lot of products contain this product. We have hibiscus. I remember hibiscus from time when I was here as an infant, so we were used to eat it, and it's sweet, and there is a high demand for hibiscus. And it's also a natural colorant, and it can improve using as a flavoring. There is a problem of supply because the cosmetic industry as well, they're looking for the hibiscus. Lycopene, which is from tomatoes. Alma, I think this one is from India, more in India. So we have seen many products that contain in this one. so this is just to give you some example of some where non-traditional. I call them non-traditional, but they're high potential there. My last part is I'm trying to show you how the interdisciplinary areas can be important. This is an example of a project that I did with ICBA, which is plant science and food science, right? Plant science and food science. And it's about quinoa. And there are a few of your colleagues here that are interested in quinoa. So as it has been mentioned, quinoa production increases, but the demand is increased and the price has increased dramatically from 2016 to 2013, nearly tripling. So it's a high, very, very high demand. But the challenge with quinoa is the agriculture practices, the genetic variability, the agronomic condition, the type of processing, the post-harvest condition, the environmental condition. All this can affect the chemical profile, can affect the nutritional value, can affect the techno-functional properties. And there is a lot still to learn from this. So if we look at the chemical composition, you can see that there is a variability here. Like if we look at the total dietary fiber, we can go easily from 7 to 15. If we look at the protein as well, there is a variability. The ash, there is a variability as well. So what we try to do, I'm not trying to convince you that quinoa is good because of the protein, of the quality of the proteins, but I want to show you some results in which we had grown quinoa in different regions with different salinity here, 2.3, 16.3, and 18.9, and we looked at their composition profile. And you can see from these results that protein content vary a lot depending on the region and the salinity as well. You can see as well that the total dietary fiber, ash as well. So the nutritional aspects we cannot only consider it at the end. It's something that should be considered from the beginning. If we look at the amino acid, remember methionine and phenylalanine, they are very important one. They had decreases when we have a high salinity. And also the ash composition, calcium, sodium, iron, we see a variability depending on how is the salinity and the soil. What we have done as well is that we did set up an experimental design in which we have varied the fertilizer treatments and the genotypes and the line. And we tried through different, that was like more than, I think the table is not complete, but we looked at the composition of quinoa that has been grown in different conditions. And we tried to understand how the fertilizer and how the genotype did affect the chemical compositions. You can see here from the protein, as it was expected, as we increase the fertilizer, we increase the protein content. The ash, there was no really variability here. However, the total carbohydrate, we see it doesn't vary. But if we break it down into total dietary fiber and starch, we see that there is a variability. So the lesson to be taken from here is the agriculture practices, the end product, the chemical profile, all these are, it has to be connected somewhere, because that's the lesson to be taken. So my conclusion, so there is a really increased interest in generation of ingredients starting from fairly simple components. And that's what was my philosophy, is to start with bioproduct, to start with the product that is abundant, and with the idea to increase the value of this product. It has been mentioned algae, and we produce a lot here in Morocco, so there is something to be done here. And also biocatalysis it's my field, and also there is a big potential, but it has challenges that we are now trying to address them. And of course, I couldn't do all this work on myself. So here it's all my graduate students, some of my graduate students, my collaborators from France, Spain, Acer, Agri-Food, and others. And thank you very much for your attention.
Thank you so much. I think we have room for some questions.
Thank you. I thank you, Madam, for the presentation. Especially in the first part, we'll get a good idea about what McGill University is doing, especially in the agricultural department. And I noticed there are many things that can be in common that we can work together on. I saw there the arboretum. My question on that, or my comment on that, we are also working on developing arboretum here in our experimental station. The objective for our objective is the inside conservation of some species, fruit trees. My question is, do you have any research program going on this arboretum that could, in collecting data and following the characteristic of different trees and so on? Because our objective here is to have this arboretum collect the species from different regions as a conservation objective, but also following the characteristic and the phenologic of different crops and their adaptation.
I think. Hello? Hello? So I agree with you. It's so important because we don't realize, but it's so important to have this database, either for insects, for plants, or for others. We do have one, and it's a challenge to maintain it. We had someone who had, his research was linked to that, where she was trying to look at the evolution of the plants, and she has all the different, all the samples are kept and everything. But she retired, but there is a technician taking care of that. So I can put you in contact of the person that she's taking care of that, but it's something that I would highly recommend because it's so useful.
Yeah, okay. The second thing is about this ag-economics department.
Yeah, agricultural economics.
Yes, we also, because the agricultural economics is very important in developing agriculture in this, especially if we add rural sociology. Right. And we, in our plan, also to develop this agricultural economic, and we may look into this collaboration to get our build up in a good way. Mm-hmm. But we need to add this social rural sociology.
Yes.
Because once we're talking about farming, we're talking about rural areas, and these people can help to identify the problem of transfer technology and so on, and also orient researchers in doing their research.
And in any cases, I think, I believe strongly in many international projects, you need to have a social and agricultural economics. Right. So it's important.
Okay. The following point is about this, I don't know about the ag mechanization in McGill University. Do you have any department of the mechanization, developing small mechanization and so on?
We have a bioresource engineering department, and we have two colleagues that they are working in mechanical, they do prototypes, like small machinery that helps because they were able to secure funding for food security. So they work with other countries, and they had helped them to develop prototypes.
Okay, and about quinoa. Quinoa is very important crops, and I believe ICBA has a project that will be initiated here for the rehabilitation of mine site using quinoa, and our university will be involved.
Yes.
But still, I agree on this genetic part of it and making more for a wide area. But we will initiate a project with ICBA here in Morocco, in Safir, in this area, with the objective of the rehabilitation of this mine site, and also get some output out of it.
Right.
The last point is about Africa, and I believe our university oriented with Africa countries and so on. I saw that you have a lot of links of different countries of Africa. Maybe there's a good way to.
I think so. I mean,
especially we're looking into the nutrition effect and so on.
They don't have to come to Canada to look for it. They have just to look at the north of Africa. There is a big potential there.
Okay, thank you.
That's something sure.
Thank you.
Thank you for the beautiful presentation.
My pleasure.
My question follows his, because I'm going to talk a little bit about Africa. There's one point you mentioned about bringing industry to the projects.
Yeah.
And one of the challenges I think what I've observed is about industry feeling that they're going to close shop because they're not getting enough ingredients or raw materials from agricultural, platform. I don't know, could you share your experience about how your research is more driven by industry and more the private sector? And how much of your research has inputs in terms of funding from private sector? Or is it a trend you see going up, or how exactly is that going?
We're trying to increase the industrial partnership. It's our goal. But this being said, we need to keep fundamental research. So we have some programs that they fund only the fundamental research. But when you are dealing with agriculture faculty, we're dealing with fertilizer, we are dealing with chemistry, it's quite applied. That's my opinion. So it's important to have the industry partnership. It's a challenge for us, but as I mentioned, I think you were not there, we had created an office, MUBEC. And these people, they have been working in the industry. It's only 2 people that they are serving all the university, but they know how to deal with industry, how to speak with them. So then you tell them, okay, I'm looking for this type of industry. They will go look and try to find the best one that fits with your need. But at the same time, what we were doing, we invest. We bring industry in our faculties, we organize workshops, we try to understand where their needs. My recommendation would be to let any project to be driven by the industry, because you have to, you cannot do it. I mean, before when I took this office, many industry will come to the faculties, Nestle and this. Okay, they come to visit, of course, courtesy, you ask people to come to present their work. And we said, we have to stop to do that because it doesn't, it's waste of time. So they have to come the other way around. So the industry, they have like a challenge, they have a need. They need to explain to us what is the need, what is the challenge, and then we find the right expertise. But when I talk about industry, it doesn't mean only big industry. There are few programs that can help even the small PME, even really the small, the women that you want to start a business. There is a potential there that can start. So building infrastructure, not only research, but infrastructure as well, that can be used by the small farmer, by the small entrepreneur, it's important because it can help them to start. And once they start and they become famous, they will think about you. And there is a real story. There is one lady, she was making gluten-free cookies to her son that he has gluten-free, the gluten-free problem, the gluten tolerance. And then her son tell her, this is so wonderful product. And she decided to come and in the pilot plant and to produce small amount. She started to sell that to small school, and now she had a company of more than 100 employees. So sometimes small ideas can be bigger. So, I mean, when you build any structure, you have to look to apply these important applied research, fundamental research, and entrepreneurial spirit as well.
Yeah, thank you very much for this very nice lecture. I have a couple of questions just for my own understanding, since it's not very clear for me. For the first part, yeah, you said that you use enzymatic reaction to selectively extract one oligosaccharide for a mixture of oligosaccharide. Is it correct?
Which part?
For the first one.
Which is the potato protein?
Exactly. You extract one of them, entire one of them?
So the first part was to isolate rhamnogalacturonal, which is a big polysaccharide with 300 kilodalton. Yes, the objective was to isolate it intact. Yeah. But after that, I used enzyme to break it down. But when I break it down, I didn't have only one more oligosaccharide with one defined unit. It's all the time a mix.
Yeah, so my first question, do you use one enzyme or a mixture of enzyme? And the second question is, which kind of transformation this enzyme induce? Is it hydrolysis? Since if it's the case, it's very difficult to distinguish between a lot of, for example, acetyl group in different sugar. How can
so
Thank you very much.
I'm using a multi-enzymatic product because I'm thinking about the applied aspects. So we have 3 criteria: to use chip enzyme, multi-enzymatic, to use enzymes that they are food grade, and also to use enzyme that will provide specificity and selectivity. They're quite selective, and that's what is nice with the enzymes, is that they can be selective to what you are targeting. So if you remember, I show a table with different multi-enzymatic products, and we had to characterize them all because I know which activity do I need. So we looked at all these activities, and then at the end we had eliminated many of them, and we kept only the one that we think is potential based on the understanding of the structures.
Okay, thank you very much.
Pleasure.
Well, thank you, Madam Serwa, for this rich presentation. Actually, you work a lot on potato, corn, and wheat, because probably these are the main crops in Canada. But in our environment, within the university, we have 3 fruit trees that are very important that can be adapted to the area, mainly argan tree, olive tree, carob tree, and also cactus. And we have right now 2 postdocs. One of them is working, Muna Al-Kharasi. She's over there, she's working on argan tree. And then Muna Al-Amawi and Yusuf Al-Kharasi is working on cactus. And one of our doctorants will go to Canada for working on carob. So there are some studies that show that these products or these crops contain some beneficial molecules. But I think the use of your tools will enhance the extraction of some essential element or essential products, and we would like it to be part of this project in order to select the most important species that Sir Mohammed Al Gharis was talking about in our in-situ collection at the farm level. Thank you.
Yeah, my pleasure. Well, I would have loved to work with the arganier and cactus and all this, but you know, when it's important to select the crops that they are in the country, otherwise you don't get funding.
Merci pour l'invitation, merci pour l'excellente présentation. Je suis Rachid Abou Taïeb de l'INRA de Satat, l'Institut national de la recherche agronomique à Satat. Je vais commencer mon intervention par le dernier titre par lequel vous avez terminé votre présentation, Effect of Condition on Nutritional Quality. En effet, les conditions environnantes impactent la composition alimentaire. Et quand on ne connaît pas la composition physico-chimique de l'aliment, on ne peut pas concevoir un bon régime diététicien ou un bon régime alimentaire. Le problème qui se pose dans la majorité des pays en développement, c'est qu'on n'a pas des tables alimentaires qui caractérisent nos produits et nos aliments. Et par conséquent, on ne peut pas prévoir qu'est-ce qu'on mange, qu'est-ce qu'on ingère et qu'est-ce qu'on a besoin et qu'est-ce qu'on aura comme carence dans notre population. Ça me renvoie à un programme national, le programme national de la lutte contre les carences en micronutriments qui a été lancé ici au Maroc et qui a concerné 3 produits principaux, 3 produits de grande consommation, à savoir le sel qui a été fortifié en iode, l'huile végétale qui a été fortifiée en vitamine A et D3 et la farine qui a été fortifiée en fer et vitamine B. Le problème qui se pose, c'est d'une part, comme j'ai dit tout à l'heure, on n'a pas des tables alimentaires qui caractérisent nos aliments. Et d'une autre part, il y a des aliments qui sont solides, comme la farine par exemple, et qu'on fait appel à un prémix qu'on ajoute. Et sur le plan technologique, c'est très compliqué de mélanger un produit solide, un prémix solide, avec un produit alimentaire solide pour avoir une fourchette ou une fourche de concentration dans l'ordre de ppm. On sait très bien que ça a posé problème, et sachant très bien que certaines personnes qui ne tolèrent pas le fer, par exemple, qui sont atteintes de la thalassémie, peuvent avoir ce genre de problème d'intolérance du fer. Donc, je ne sais pas, dans votre université, est-ce que vous vous êtes attaqué sur ce genre de recherche ou pas? Ça, c'est la première question. Deuxième question, j'aimerais bien savoir si, au niveau du Canada, et principalement concernant l'ACIA, l'Agence canadienne de l'inspection alimentaire, est-ce qu'elle s'est penchée sur la création la création d'une certaine conscience en matière d'additifs alimentaires? On a une vraie confusion on a des consommateurs. Bon, la majorité des consommateurs ne peuvent pas déchiffrer ce qui est écrit, ce qui est mis sur l'étiquetage. Par exemple, ici au Maroc, on a une grande confusion entre la tartrazine et la curcumine. Quand on consomme le E102, la tartrazine, qui est extrêmement allergène, comme kharkoum, c'est grave. Donc, je sais pas, c'est un point que je pense assez important et qu'il y a une vraie relation, une relation qui est très étroite avec notre alimentation, notre régime et notre santé, bien sûr. Et j'aimerais bien avoir une idée sur les recherches qui ont été menées dans votre université et pourquoi pas au Canada en général. Okay. Merci.
Merci. Alors, pour revenir à la première question sur la fortification des aliments. La fortification, je pense que maintenant, si vous vous rappelez, j'ai montré un slide, j'ai parlé des plusieurs générations. Là, on est plus dans la génération 2, où on fait plus de fortification, mais on cherche à intégrer ou à incorporer dans les produits des composés bioactifs. Là, on a des jus d'orange avec fish oil, oméga 3. Donc, il y a beaucoup de choses qui se font à ce niveau-là que de la fortification. Puis, ça reste un défi. Le mélangeage, le mixage, je pense que c'est un défi agroalimentaire qui est très important. Et comment s'assurer que la consistance d'un batch à un autre, et puis dans tout ce qui en suit. Maintenant, quand on parle de labeling, il y a eu beaucoup de confusion. Mais les consommateurs... Okay, I'm going to switch in English. So the consumers are really driven the trends, and there is a high demand for clean label. It's amazing now. Clean label, which is no chemicals, and shorten the list as much as possible. So this is the demand from the consumers, and many companies, they have to adapt. Just to give you an example, so if we talk about the processed meat, what they're doing now, they're using freeze-dried vinegar. Freeze-dried, and then they added and they put freeze-dried vinegar, which is an acetic acid, and so we're using a lot of extracts, garlic extracts, vinegar, and many other ones as an anti-preservatives, and antimicrobial components to be able to ensure the clean labels. But it's still a big deal. Now, the most important aspect is to be transparent with the consumers. The consumers, you have to tell them when it is in product and it's their choice to select it or not. And what we are trying to do, if we talk about the curcumin, is to try to modify the curcumin naturally by fermentation or by other things. Then when you incorporate it, it will not affect the taste, it will not affect the color, but still natural. So I don't know if that answers your question. Yeah, the confusion is there. I mean, how can you do it? It's there, and regulation. The way how we regulate foods in Canada, we have what we call a food identity. So when you define a product as a milk, it has to have a certain composition. If it doesn't have the certain composition, you cannot label it as a milk, bread, and so on. And we have a list of food additives that they are permitted. You can use only this list of food additives. And if you add any new additive, it has to go through all the process. But we have the food ingredients, which we isolate. Food ingredients are isolated components from food, like used protein, soya protein, rice protein. It's a food ingredients. You can use it. You don't need to have the list. But I think what has driven that is the consumers. Consumer wants clean label. Most companies now, they're trying to go with the clean label. No chemicals, less additive, shorten the list. That's their challenge.
Yes, thank you for showing first that the benefits of using biocatalysis instead of organic chemistry to come up with this oligosaccharide and use it for prebiotics. Thanks for that. My question is about how this translates to industrial processes and bioprocesses, and what's the challenges today of using all this enzyme within an industrial pilot and keeping the yield that has been demonstrated in the lab, in the bench lab. Thanks.
Thank you. So the biocatalytic processes are used in pharmaceutical company extensively. Stop, no? It's working? Okay, sorry. So it has been used extensively because we are looking into the active. We are dealing with enantiomer and all that, so that's the only way to go. But it is also used in the food industry, like when we want to remove pectin from cranberry, they use pectinase and so on. But I agree with you that the challenge is to be able to develop a process that is economically viable, which has been all the time the challenge. I didn't show the example in which we have trying to use a solid reaction with enzymes. And I like this project in which we are trying to avoid many downstream steps and to be efficient as much as possible. We are immobilizing enzymes. So there are many things that can be done to improve it, but it doesn't mean it's biocatalysis. I think it's already there in food, in agri-food.
Yeah. The second side also of my question is, so what's the scientific foundations proving that these prebiotics are really beneficial for men?
Yeah.
Since it has been demonstrated for animal very well, but today we haven't yet heard about any clinical investigation of.
So the European community did shut down everything. The European community decided that they cannot label their product as good for intestinal health and prebiotics.
True.
And I think the reason why, because when you want to mention any health benefits, it should be a direct effect, which is very difficult to prove because you have a specific bacteria that is able to ferment, that you have this linked. But at the same time, I know in University of Brussels and a few other ones, they're working actively in finding this link and proving it. So it just time.
The example is of Danone, who was talking about the bifid is active.
Yeah.
And then he just removed.
Right. So
thanks.
Pleasure.